everyone, I am Dr. Sonal Jain from Department of Anthropology, University of Delhi. In this series of forensic science lecture, today we are going to discuss about different type of centrifugation techniques. When we talk about the centrifugation techniques, they are based upon what kind of evidence you have found out the crime scene and what do you want to extract out of it. Either we have to separate, either we have to purify certain substance, either we are looking for certain specific types of cell. So, depending upon that, we use different kind of centrifugation techniques. The most important centrifugation techniques which we are using are density gradient centrifugation, differential centrifugation and ultracentrifugation. When we talk about the density gradient centrifugation, it is divided into two parts: rate zonal centrifugation or isopycnic or sedimentation equilibrium centrifugation. Density gradient centrifugation, as the name suggests, it allows the separation of many or all components in a mixture and allows for measurement to be made. Two types of density gradient centrifugation are there: rate zonal or isopycnic or sedimentation equilibrium centrifugation. This type of centrifugation is used to purify viruses, ribosomes, membranes, etc. In this, a sucrose density gradient is created by gently overlaying lower concentrations of sucrose on higher concentration in the centrifuge tube. So, as we have discussed the density gradient practical for the soil identification or for the glass particle identification in our soil lectures or in our forensic physics lecture. So, similarly, such type of arrangement is being made in the centrifuge tube, where the different layers are being made with the sucrose having different concentrations. So, depending upon the density, different types of viruses, ribosomes, membranes which we are trying to separate or purify, they will come as a layer in the gradient. So, the particle of interest are placed on top of the gradient and they are centrifuged in ultracentrifuges. The particles travel through the gradient until they reach a point at which their density matches the density of the surrounding sucrose. This fraction is then removed and analyzed. So, basically we have made a gradient inside the centrifuge tube and the particles are separated based on their densities and they have taken their place in the tube. Whereas in the rate zonal centrifugation, the solution has a density gradient, the sample has a density that is greater than all the layers in the solution. So, the sample is applied in a thin zone at the top of the centrifuge tube on a density gradient, and when we apply the centrifugal force, the particles will begin sedimenting through the gradient. So, as we can see in the diagram, that we have the various layers of different densities. So, depending upon the density of the particles, they will take the layer as per their density and they will begin sedimenting in the separate zones according to their size, shape and density. We have to be very careful when we are extracting or purifying or separating or sedimenting the particles based on the rate zonal centrifugation. We have to very carefully pipette out the layers which are of our use and then take the different tubes and then separate using where is a clear cut mark of different densities where the particles are being separated. However, in the zonal centrifugation, it is also known as the band or gradient centrifugation, as we have seen that different bands are there and different zones are there where different particles are being lined up based on their property of density. It also relies on the concept of sedimentation coefficient, that is movement of sediment through the liquid medium and separating based on their densities. In this technique, a density gradient is created with sucrose and high density at the bottom. The sample of protein is placed on the top of the gradient and then centrifuged. Whenever we are doing such kind of experiment, we will place the sample on the top. The protein sediment according to their sedimentation coefficient and the fractions are collected by creating a hole at the bottom of the tube. That is mostly used when we are using or working with the proteins. In such cases, we made a hole at the end of the tube and then we can collect the sample at the bottom. So, in the rate zonal and the isopycnic sedimentation, we can divide based on the different zones where we are separating it on the basis of the density. Wherever in the isopycnic we are making the density gradient with a sucrose layer and the separation is being happening depending upon which layer is being taken by the sample. So, rate zonal and the isopycnic, they both come under the density gradient centrifugation, and depending upon what kind of separation is being required, what is the basic criteria we are looking for, what kind of materials we have to purify, we can either choose the rate zonal or the isopycnic centrifugation. In this type of centrifugation, the solution contains a greater range of densities and the density gradient contains the whole range of densities of the particles in the sample, where each particle will sediment only to the position in the centrifuge tube at which the gradient density is equal to its density. That is why we call it as equilibrium centrifugation. In this equilibrium centrifugation, as we can see that the separation of particles occurs into zones on the basis of their density differences, which is independent of time. So, whenever we have a mixture, the substances, they are of different particles are of different densities, a centrifugal force is applied and these particles come in equilibrium with their zone having a particular density. So, they are being separated based on that. This is also called as density gradient centrifugation. The solution of the biological sample and cesium salt is uniformly distributed and rotated in an ultracentrifuge. Under the influence of the centrifugal force, the cesium salts redistribute to form a density gradient and the sample molecules move to the region where their density equals to the density of gradient. This is different from the differential centrifugation, which is a common procedure used in microbiology and cytology, used to separate certain organelles from whole cells for further analysis of specific parts of cells. So, basically in differential centrifugation, it is based on size, shape, etc., whereas the density gradient is typically based on the density of the particular materials. So, in the differential centrifugation, a tissue sample is first homogenized to break the cell membranes and mix up the cell contents. The homogenate is then subjected to repeated centrifugations and each time removing the pallet and increasing the centrifugal force. So, this is in our hands how we are using the centrifugation technique. Supposedly, we can control the speed of the centrifuge, we can either increase it or we can either decrease it. We can have repeated centrifugations depending upon what we are looking for. Each time we can remove the pallet and each time we can remove the supernatant depending upon what kind of material we are using and the centrifugal force we are applying. In the differential centrifugation, this is mostly used by the biochemist, where tissue such as liver is homogenized at 32 degree in a sucrose solution that contains buffer. This homogenate is then placed in a centrifuge and spun at a constant centrifugal force at a constant temperature. At a certain time, the sediment forms at the bottom of the centrifuge called a pallet and the overlaying solution is called supernatant. The overlaying solution is then placed in another centrifuge tube which is then rotated at higher speed. So, basically one material which is being extracted in one tube where we have supernatant or pallet, that can be then further transferred into another tube which can be then further rotated to extract the other substances or the other particles which we are looking for. So, depending upon we can go different types of, after the first step we can have the low speed centrifuge, then we have a pallet there, then we have one supernatant, then we have the pallet 2, then we can remove the supernatant. So depending upon, we can, until we reach at a point where we are looking for a specific kind of pallet or a supernatant, we can have the multiple rounds of centrifugation depending upon the increased speed of that, depending upon the lowering the speed of that, changing the buffer, which will help us in achieving at the perfect stage or the step where we are looking for a specific cell, specific organelle or a specific nucleic acid. So depending upon, either you are extracting DNA out of the biological material or extracting RNA out of the biological material, the buffers will differ, the revolution per minute speed will differ, the centrifugal force will differ depending upon what your target is to. Now another important technique here, it is the ultracentrifugation, where it is involved by, applied by Swedberg to any centrifuge that permitted observation of the contents of the container during the act of centrifuging. So if we are going for the analytical ultracentrifuge, we have to look for, supposedly we want to study the sediment behavior of the particles, then we have to look for how they are behaving. So in the ultracentrifuge, we can apply a microscope, a detector, which can let us in knowing that how the particles in behaving, what is their sedimentation behavior. Nowadays the ultra high force centrifuge can go up to around 75,000 rpm with the relative centrifuge force up to 5 lakh Gs. Swedberg only coined the term ultracentrifuge. He was a colloid chemist. He used it is to determine the MW and subunit structure of hemoglobin, studied that changed the ideas concerning the structure of proteins. The first commercial ultracentrifuge was produced in the year 1940 by a company called Spinco. So this ultracentrifuge, when it is applied with an analytical technique, the sample is being spun and can be monitored in real time through an optical detection system using UV light absorption or an interference optical refractive index sensitive system. So basically what we are doing in such cases, we are involving two instruments together. We are using one absorption detector system and one centrifuge, and then we are looking how the particles is behaving when we are using this analytical system. This system allows the operator to observe the sample concentration versus the axis of rotation profile as a result of the applied centrifugal field. Basically this type of analytical centrifuges, they can be used in certain cases where we are doing novel researches, where we want to know how the different particles, what is their sedimentation behavior, how they are behaving with the applied centrifugal field. Then we have to look for certain different particle size or the different substances. Whereas when we talk about the preparative ultracentrifuges, they are available with a wide variety of rotors which are suitable for a great range of experiments. The rotors are designed to hold tubes that contains the sample. As we discussed about the fixed angle rotator and swinging bucket rotator, they allow the tube to hang on the hinges so that the tube reorient to the horizontal as the rotor initially accelerates. These rotors are used in biology for pelleting of fine particulates, fractions such as cellular organelles, mitochondria, microsomes, ribosomes and viruses. So, the Theodor Swedberg, who invented the ultracentrifuge in 1923, he won the Nobel Prize for the same in 1926 for his research on colloids and proteins using the ultracentrifuge. The ultracentrifuge machine is very expensive and it is a limited time. After that, we have to get a new machine. This is an important tool in biochemical research, which through rapid spinning imposes high centrifugal forces on the suspended particles or the molecules in the solution, which causes the separation of the matter on the basis of differences in their weight. So, this ultracentrifugation works on the principle of the differences in the weight. So, let us take an example of red cells separated from plasma of blood, nuclei from mitochondria in cell homogenates, one protein from another in complex mixture, and also isolation of macromolecules such as DNA, RNA, lipids, etc., where the rotational speed is up to 1,50,000 rpm, which is creating a centrifugal force up to 9 lakh G. Such a high speed and a high centrifugal force can be achieved by ultracentrifuges, and for that we also have to maintain a proper cooling system, because such kind of centrifugal force, when it is appearing, can lead to the heat generation, which can help us in distorting the process of the machine. The two types of ultracentrifugation is the analytical, where the interactions between micromolecules can be analyzed, and the another one is preparative, where we can isolate and purify specific particles such as subcellular organelles. So, in analytical centrifugation, two types of experiments can be performed: sedimentation velocity experiments, where they interpret the entire time course of sedimentation, report on the shape and molar mass of the dissolved macromolecules as well as their size distribution. Whereas another type of experiment which can be done is sedimentation equilibrium experiments. These are concerned only with the final steady state of the experiment, where sedimentation is balanced by diffusion opposing the concentration gradients, resulting in a time independent concentration profile. Preparative ultracentrifugation is to isolate specific particles which can be reused. It can be of different types, like differential ultracentrifugation, which is a common procedure in microbiology and cytology, which is used to separate certain organelles from whole cells for further analysis of specific parts of the cells. Density gradient, as we have discussed, can be zonal or rate, and it could be isopycnic. In the zonal or rate centrifugation, the mixture can be separated. On the top of a gradient, the sample is being put, and the increasing concentration down the tube is present over there, and as the sample moves down the tube at different rates, it can be separated. Whereas in isopycnic centrifugation, the same density we have to maintain, and the molecules are separated at the equilibrium position, where each molecule floats or sinks to a position as per the density of the particular material. So, in the ultracentrifuge, the analytical type, when we discuss, we have the cooling chamber, we have a vacuum pump, UV light source, we have motor, rotor, we have a chamber and mirror, light path and detector. This is basically when we are doing SVE study or SEE study, which we just discussed, that we are looking at the macromolecules, how the macromolecules are behaving in the ultracentrifuge, that we look using the analytical ultracentrifuge technique. So, this analytical and preparative centrifuge, they have different functions. For analytical, they use small sample size. They can have built-in optical system to analyze the progress of molecules, and during centrifugation, they use relatively pure samples. They can be used to precisely determine the sedimentation coefficient and molecular weight of the molecules. The most common example is the Beckman model, which can be used for such purposes. Whereas, if we go to the functions of the preparative centrifuges, the larger sample size can be used. This does not have any optical readout system. We can use the samples which are less pure, and we can estimate the sedimentation coefficient and the molecular weight. It is generally used to separate organelles and molecules, where the most centrifugation work is done using the preparative centrifuge. It also have the ultracentrifuge can have 4 different type of rotors: fixed angle, swinging, vertical and near vertical. Fixed angle and swinging bucket, which we have just discussed, which were also there present in the differential centrifugation, where the rotors, they are usually made up of aluminum or titanium. A titanium rotor is designed by T or TI, whereas the composite rotor is designated by C, for example VC53. So, the different types of rotors, when we are discussing, they can be made with the pure aluminum or it could be an alloy. Titanium rotors, when we talk about, they are very strong and chemically resistant than aluminum rotors, but they are quite expensive. These buckets and lids are high performance rotor and they can be usually paint red for identification. When we talk about different types of rotors, we have fixed angle where it is being fixed from 25 degree to 40 degree Celsius. This 20 to 45 degree, the axis of rotation is fixed. So, depending upon that, we use the fixed angle. Where swinging bucket we see, they are used for the piloting or isopicnic studies. Tubes are attached to the rotor body by hinge pins or a crossbar. The buckets swing out to a horizontal position. So, when isopicnic studies are done, as we can see in this type, the swinging bucket goes up to the 90 degree, and then on the basis of the sedimentation, there can be different zones where the particles are sedimented. The vertical rotors, they hold the tube parallel to the axis of rotation, therefore bands separate across the diameter of the tube rather than down the length of the tube. And this vertical rotor, you can see in the diagram, they are useful for isopicnic, and the bands will be in the axis of rotation will be same as the similar, whereas in the swinging bucket it goes up to the perpendicular 90 degree. Near vertical is, they are basically designed for gradient centrifugation where there are components and that do not participate in the gradient. So, basically they are not vertical properly and they are not fixed angle. Their angle is between 7 to 10 degrees and to the axis of rotation. In this rotor used only in quick seal and opti-seal tubes. The most important thing when we are doing the centrifugation, we have to balance the rotor. The mass of a properly loaded rotor will be evenly distributed on the ultracentrifuge drive hub, causing the rotor to turn smoothly with the drive. Depending upon 2 sample, 3 sample, 4 sample, 6 sample, they should be evenly balanced. If our rotor will be unbalanced, it will not rotate properly. The balanced rotor rod fill all opposing tubes. Weight of opposing tubes must be distributed equal. It's not like we will put a tube in front of that, but if a 5 ml tube is put with a 5 ml liquid, then the opposite should always be 5 ml. So, the opposite band should always be filled and with the same amount of the material which is present on the opposite side, and their tubes always should be placed symmetrically in the rotor. So, to start a run, we set the RPM, time and the temperature. Never exceed the manufacturer's starting maximum speed. Press the start button and wait until we reach the desired speed. So, we put the exact time which we have to rotate for, the speed we are rotating it, and the temperature if it is a temperature control centrifuge machine. After the spin, when the run is completed, never attempt to open the door or slow the rotor while the rotor is in motion, and whenever we are using it, we have to log in for further maintenance and repair. A logbook should always be maintained with the centrifuge, and the rotors after using, they should be properly cleaned. They should be washed and use a cleaning kit for any radioactive materials if present, and never leave an unclean rotor to dry out, which will damage the rotor. We should sterilize it with 70% ethanol. We should always open the centrifuge door, install the rotor. We should always properly follow the manual which is being given by the expert provider who has the centrifuge machine manufacturer, and we should always carefully read the operating manual before following that. So, the takeaway points is the centrifugation technique is the best separation technique which can be used in all the biochemical and biological tests. But the important thing is we have to take care of the centrifuges and rotor so that they work properly. And to conclude, I would say the centrifugation, the different type of centrifugation is one of the important techniques which can be analyzed and used for different types of criminal evidences which are found at the crime scene. Thank you.